Hello, everybody. I hope each one of you is keeping well and including your families. And I hope you're all making the most of these online lessons. Okay. So, as you know, we are currently discussing the chapter Human Reproduction. Now, before we move any further, I would like to give you a chapter overview. We do this every time so that all the different segments which we are going to look forward to, they get registered in your mind thoroughly. We have already discussed about the male reproductive system and the female reproductive system, as you're aware of, correct? Then the next aspect we need to discuss is the formation of gametes, that is gametogenesis, formation of sperm in case of male and egg in case of female. Then we need to discuss about the cyclical changes associated with the female reproductive system in primate mammals, which is called menstrual cycle. Then following that, the formation of zygote, fertilization and attachment of the embryo to the uterine lining, that is implantation. Then coming to pregnancy and development of the embryo. And lastly, we have childbirth or parturition and production of milk or lactation. So this is just to give you a roadmap as to where we are headed to in this particular chapter, okay? Now, before I start with anything, as you know the drill. It's about a quick review as what we had learnt in the previous lecture. So in the previous lecture, we had discussed about the female reproductive system with a lot of emphasis laid on the diagrammatic sectional view, to be very precise, the anatomical aspect of the female reproductive system. Yes, you all agree with me, right? I am assuming that you all have practice the diagram well. You have written the homework satisfactorily, and you have learnt the concept thoroughly from NCERT and question bank, and you all at the end of the day are confident with the syllabus. Okay, then we discussed about the structure of the vulva or the external genitalia, and then we moved on to the mammary glands. The mammary glands are structures which are closely integrated with the female reproductive system, functionally very closely integrated with the female reproductive system. So this was what we had discussed in the previous lecture. Now in this lecture, what we will be discussing about specifically is a process referred to as gametogenesis. So what is gametogenesis? Gametogenesis is the process of production of gametes. See, the name itself indicates gameto refers to the gamete or the sex cell. It's sperm in case of male and egg in case of female, and genesis refers to its origin or its production. So how do you define the term gametogenesis? All of you, please bear in mind, this is a very important 1-mark question for your board exam. So I hope you're all sitting with your pen and with your book open. I want you to jot down the definition of gametogenesis very quickly as I'm reading it out to you. So obviously it is the production of sperm in case of male and ova in case of female, sperm production in the testis and egg production in the ovary. And this process of production itself is referred to as gametogenesis. All right. To be very specific, it is referred to as spermatogenesis in case of male. It is referred to as oogenesis in case of female. To create a distinction between the two, spermatogenesis in male and oogenesis in female. So I'm repeating the definition for all of you. I'm assuming you are jotting it down, and please try and register the definition. It is the production of sperm and ova by the testis in male and ovary in female respectively. So by this you can conclude that the site of production of sperm is the testis and the site of production of ova is the ovary. Clear to all of you the definition? Okay, now in the next slide we will be turning our focus on to specifically the process of sperm production. Now, this process which specifically deals with sperm production is referred to as spermatogenesis. Now, as you must have guessed, it happens only in the male sex. And where does it occur? In the testis. Isn't it? The male gonad or the testis? Yes, you're right. How do you define the process of spermatogenesis? Again, this is a very important 1-mark question in your board exam. and I want all of you to once again note down the definition of spermatogenesis. It is the process of production of sperm by immature male germ cells, and the technical term for immature male germ cells is the spermatogonia. And this process happens in the seminiferous tubules of the testis, and it begins at puberty. See, one definition has so many important components, and if you miss out on any one of these components, then your answer is incomplete. That's why definitions are very important, and 1-mark questions do not have a choice in your board exam, and that's why I'm stressing upon this. You all have to be very thorough with 1-markers like this. So how do you define spermatogenesis? It is the process of production of sperm by immature male germ cells called spermatogonia, happening in the seminiferous tubules within the testes. And when does it begin? It begins at puberty. Now I want you to remember this particular aspect, okay, that is the process of spermatogenesis begins at puberty, because when I discuss with you all the process of egg formation in female, that is oogenesis, I will be discussing about the timeline, when does it start and when does it progress and such. So here you should remember that spermatogenesis begins only at puberty. Is that clear to all of you? So did you take down the definition? The process of production of sperm by immature male germ cells in the seminiferous tubule of the testis at the time of puberty. Okay, fine. Now let us move on to the next slide. Now we are going to discuss about the actual mechanism of spermatogenesis. Now I want all of you to give the heading in your books as the schematic representation. Now this is the schematic representation of spermatogenesis, and this will be asked for a 3-marker question in your board exam. Sometimes they may ask you to explain. So you will have to draw the schematic representation and you will have to explain it in words. So when the question has asked for an explanation as well, it is mostly a 5-mark question. If they ask you only the schematic representation, it is a 3-mark question. Many of you commit the mistake of not reading your question properly in your examination, because sometimes they would have asked with the schematic representation explain, and some of you just end up writing the scheme and just leaving it at that. So then you will end up losing 2.5 marks, isn't it? So read the question carefully in your examination or when your homework questions are given to you, so that your answers are accurate and it caters to the question that is asked. Okay, so now let us move on to the process of spermatogenesis. So the first step, it's already mentioned here that the process of spermatogenesis happens when a person becomes sexually mature, when the male becomes sexually mature, when the male attains what is referred to as puberty. Correct? So the first step is, I'm highlighting this, I'm putting a flower bracket here and I'm writing 1 over here because this is the first step in spermatogenesis. Notice these cells which are referred to as the spermatogonia. The spermatogonia are nothing but the immature male germ cells, and they are dividing repeatedly by what division? By mitotic division. Did you understand? So this first stage of spermatogenesis involves continuous mitotic divisions of the immature male germ cells, which are referred to as, what are they referred to as? Spermatogonia. Yes, you're right. So here they have shown how this one cell is dividing into 2 cells, and each of them are dividing into 4, and one of them again is dividing into 2. Correct? So this is what is referred to as the phase of multiplication, the first phase of spermatogenesis, where there is rapid multiplication by mitosis, can be called as the phase of multiplication. However, they have not distinguished such phases in your NCERT, so I will not be stressing more upon it. But the take-home message is there is a lot of mitosis happening here. Okay. Now, I want all of you to focus on this one cell. Now, this cell is referred to as a primary spermatocyte. Okay. Now, you may ask me a question. How is this cell different from spermatogonia? Now, what you need to remember about this cell is this particular cell has now entered, I want you to write down, has now entered meiosis. So it decides, I'm done with mitosis. I don't want to divide by mitosis anymore. I'm tired of mitosis. Now let me step into what? Let me step into meiosis. Once a spermatogonium stops dividing by mitosis and steps into meiosis, you will call it as, what will you call it as? Primary spermatocyte. Did you understand what I'm saying? So this cell has stopped dividing by mitosis and it has now entered meiosis. And such a cell which has now entered meiosis is called a primary spermatocyte. All of you notice one more thing. Both the primary spermatocyte as well as the spermatogonia have a diploid number of 2n is equal to 46. 46 chromosomes in the nucleus. Please try to understand. Diploid meaning having two sets. So they have two sets of chromosomes coming up to what number? 46. Is that clear to all of you? So the first phase was mitotic division, and then after enough mitotic divisions have happened, one of them decided, I am done with mitosis, and they entered, and this cell has entered meiosis, and now we call this cell as a primary spermatocyte. Let us trace the journey of the primary spermatocyte now. Once it enters meiosis, it successfully completes, I'm writing here, meiosis 1. Please remember from your first year basics, meiosis happens in two steps. Meiosis 1 and meiosis 2. So now it has successfully completed meiosis 1, which is a reductional division. Reductional means the number of chromosomes get reduced. And it produces two daughter cells. I'm highlighting the two daughter cells for you. And these daughter cells are now called secondary spermatocytes. They are products of what? Meiotic division. Which meiotic division? Meiosis 1. Did you notice that the secondary spermatocyte has only half the number of chromosomes? N is equal to 23. How did 46 become 23? Because meiosis 1 is, I'm writing in brackets here, it is reductional. There is reduction in the chromosome number. And I want all of you to notice one more thing. Both the secondary spermatocytes are equal in size. They are of the same size. You will know why I am stressing upon this point later. Right now, just remember that at the end of meiosis 1, we have two equal size secondary spermatocytes from one primary spermatocyte. And are they haploid or diploid? Haploid. Because meiosis 1 is reductional. Clear? Is this the end? No, it's not the end. Because each of the secondary spermatocyte, now see what I'm showing you here, will now undergo meiosis 2. Please remember, meiosis happens in two steps. Now it's undergoing the second stage. Each of the secondary spermatocytes undergo meiosis 2 to form two each of cells which are referred to as, so two from one secondary spermatocyte and two from the second secondary spermatocyte, a total of 4 haploid cells which are referred to as the spermatids. Notice that the spermatid also has haploid number of chromosomes, n is equal to 23. So we ended up with how many spermatids? We ended up with 4 haploid spermatids. Is that clear? Now this marks the end of the major part of spermatogenesis. But is that all? No, that is not all. Notice what's happening to each of the spermatids. Each of the spermatids are transforming into tadpole-shaped sperm. This process of differentiation, I'm going to write it here. This process of differentiation, please take down. It is referred to as the spermiogenesis. So in spermiogenesis, some transformation happens. Transformation of what into what? Transformation of spermatid into sperm. Is that clear to all of you? So we end up with a total of 4 sperm for how many primary spermatocytes? We started off with, all of you please bear in mind, I'm going to try and link it here. We started off with a 1 primary spermatocyte, and now we end with how many sperm? 4. So the ratio is 1:4. I'm writing it here for you on the left hand side. 1:4. What is 1 over here? 1 refers to primary spermatocyte, and 4 is the sperm. Are you understanding what I'm saying? Now don't worry if you're not completely satisfied with this. I'm going to draw and show it to you in the next slide. Okay? And then you can draw along with me so that you will understand the concept better. Okay? And if you have any doubt in this particular flowchart, you know that you can always rewind and watch the video again. Fine. So let me move on to the next slide. Alright, so this blank slide, I'm going to draw the process of spermatogenesis here, the schematic representation, and I want all of you to follow what I'm drawing and put it on paper so that your concept is very, very thorough for you. Okay? So which is the first cell? The first cell was a spermatogonium. A spermatogonium, or the male germ cell. See how it is dividing by mitosis here. 1 becomes 2. Okay, then 2 becomes 4. How is it dividing? Mitosis. So I'm going to label this cell as spermatogonium. Can you recall and tell me what was the chromosome number? Yes, it was diploid. 2n is equal to 46. And can you tell me what was the common name of spermatogonium? In brackets I'm going to write. It was called the immature male germ cell. So it's dividing by what division here? Mitotic division. Alright. Now let us take this cell now. This is a rebel now. Why is it a rebel? Because it's deciding I want to stop undergoing mitosis and I want to now enter meiosis. So I'm going to call this cell the rebel which decides to get out of mitosis. What should I call him? Primary spermatocyte. And he also has 46 chromosomes. 2n is equal to 46. Now he has already entered meiosis. He's done with mitosis. So let me put a bracket here and let me say it is all but, nothing but mitosis. Okay. Then now this person here, this cell here is going to divide by meiosis, and he is going to produce 2 equal sized daughter cells. And here what should I write? What division is it undergoing? It's completing the first part of meiosis, that is meiosis 1. Okay, now what should I call these 2 equal sized daughter cells? I must call them now as secondary spermatocytes. Should it also be 2n is equal to 46? No, wrong. It has undergone meiosis 1, so it should be n is equal to 23. See, 46 which was diploid has become haploid. How many secondary spermatocytes? 2 equal sized secondary spermatocytes. Correct? Now, each one of the secondary spermatocytes is now going to undergo What division? Let me put a dotted line here. And let me write on top of the line meiosis 2. Isn't it? So each of the secondary spermatocyte will now undergo meiosis 2 to form 2 each, and a total of 4 smaller cells. Actually they should be smaller in size compared to second, but all of them are equal in size. And now we call these cells which are a product of meiosis 2 as spermatids. A set of 4. N is equal to 23. Again they are haploid. Okay, I hope you're writing along with me. The flowchart or the schematic representation of spermatogenesis. Then each of the spermatids. So we have a total of 4 spermatids. What is the ratio I told you about? The ratio was 1:4. Isn't it? 1:4 ratio. One primary spermatocyte gives rise to 4 haploid spermatid. Okay. This is not the end. Now each of these spermatids, notice the spermatids are spherical. They don't have a tail. They have lot of cytoplasm. Now they are going to transform into these tadpole shaped cells. Now these are the spermatozoa, or simply let me call them as the sperm. Okay, total of 4 sperm I have now here. Now this process of differentiation or transformation, can you recall and tell me what was it called? It was called spermiogenesis. Clear? So we got a set of 4 sperm from one primary spermatocyte. Alright, so this primary spermatocyte, all of you please remember one thing: primary spermatocyte is nothing but a spermatogonium which has decided to stop undergoing mitosis. I hope you remember. Okay, so you can consider it as there is a spermatogonium. This spermatogonium, there is one of it. This becomes a primary spermatocyte. So 1 becomes 1. How did it become this? Because it says no to what? To mitosis. I am done with mitosis, it says. Now this one primary spermatocyte gives rise to 4 spermatids. And 4 of them become 4 sperm. So if you start with either a spermatogonium or a primary spermatocyte, which is nothing but one and the same, because a spermatogonium which quits mitosis is called, or which enters meiosis is called a primary spermatocyte. So we get a ratio of 1:4. So I hope this is clear to all of you, the process of spermatogenesis. You can always rewind and watch again. Okay. Now why it was drawn here is so that you are supposed to replicate it on paper. So I hope it is clear to all of you, and I hope you have drawn it in your books, because this is a very important 3-mark question. Like I said, with the explanation, it is a 5-mark question. This is how the schematic representation should look, and more importantly, you should mention the number of chromosomes at each stage. A spermatogonium has 46 chromosomes. A primary spermatocyte has 46 chromosomes. Both are diploid. A secondary spermatocyte becomes haploid, 23 chromosomes. A spermatid also is haploid, 23 chromosomes. And obviously the sperm also will have a nucleus with 23 chromosomes. So whichever cell has 46, it is a diploid cell, and a diploid cell gets reduced to possess 23 chromosomes, which will now be called a haploid cell. That's why meiosis is important. Meiosis 1 and meiosis 2. And the ratio is 1:4. Okay. And how is a spermatogonium related to spermatocyte? Now, a spermatogonium, like I said, keeps dividing by mitosis, and the moment it decides to stop mitosis and enter meiosis, you will call it as a primary spermatocyte. So this primary spermatocyte is a spermatogonium which has stopped undergoing mitosis and has entered meiosis. All right. So these are some of the minor details that you need to keep in mind, okay, before moving further. So that completes the schematic representation of spermatogenesis. The next aspect which I had incorporated into spermatogenesis itself is the spermiogenesis. How do you define, all of you? Please write down the definition. It's a 1-mark question again. I cannot stress enough about 1-mark questions because I know, especially all of you who want to get 100 on 100 in bio paper, I want all of you to be very, very, very thorough with all the definitions and everything that's given in bold in the textbook and every nook and cranny of your textbook. Okay, so fine. Now the spermiogenesis is the process by which the spermatids transform into spermatozoa. You know that a spermatid is a cell. It's a tiny cell which has a nucleus which is immotile, no tail, lot of cytoplasm. See, let me put dots for cytoplasm here, lot of cytoplasm, but then that gets transformed into a tadpole shaped structure which is the sperm. And this process where the spermatid transforms into the sperm is referred to as spermiogenesis. Okay, there's another term for spermiogenesis just for your information. It is called the spermateliosis. Maybe it's a competitive aspect, it's not in the textbook, but just because I'm talking about it, spermateliosis is referred to as is the alternate term for spermiogenesis. Now here you can see a picture, a beautiful picture showing how a spherical spermatid is slowly developing a flagella here, and how the nucleus is condensing, and how the Golgi apparatus forms a cap on the nucleus to become a structure called acrosome, which I will be dealing with in a little while. And finally it becomes a sperm which has a head, a middle piece, a tail, and so it is highly motile and it has very, very scanty cytoplasm, whereas a spermatid has excess amount of, it has how much cytoplasm a cell usually should have, but a sperm has very little cytoplasm in it. Did you write down the definition of spermiogenesis? Okay. Now here I would like to draw your attention to something which we had already discussed in the review also. A process called, I'm writing at the top of the slide for you, spermiation. You have written this in your homework. 1-mark question. Spermiation means, please take down, it is the release of sperm from the seminiferous tubules. Okay, so the sperm are set free from the seminiferous tubule and they enter into the next duct. Do you remember where do they enter next? They enter into the rete testis, isn't it? So this process where the sperm are set free and are released from the seminiferous tubules, this process is referred to as the spermiation. All right, fine. Now moving on to the next slide. I hope it is clear to all of you until here. Hormonal regulation of spermatogenesis. What are the hormones which control the activity of the gonad, the testis in the male reproductive system? Okay, now since we are talking about hormones, we need to do a little a brush up on the endocrine system. The most important parts of the endocrine system are the hypothalamus. Hypothalamus is a part of your brain. It is something which is controlling your pituitary gland. You know that pituitary gland is called the master endocrine gland. However, I would consider that a misnomer, because pituitary itself is under the control of whom? It is under the control of hypothalamus. Okay, so coming to the hormonal control, the hypothalamus, which is located in the brain, it secretes a hormone called gonadotropin releasing hormone, GnRH. I want all of you to please note down this table also in a very simple format. Okay, now let me give you the full form for GnRH over here. I'm going to write it here for you. Gonadotropin releasing hormone, GnRH. Now this is released by hypothalamus, and hypothalamus, this hormone, that is gonadotropin releasing hormone, acts on, to be very precise, it acts on anterior pituitary gland. Take down, first you draw the you don't have to draw boxes actually, just write hypothalamus and show a downward directed arrow and write anterior pituitary. Beside the arrow you can show GnRH. Are you writing? Please write it down. It'll help you register the concept better. It'll reinforce the concept in your minds. Okay, so hypothalamus releases a hormone, secretes a hormone called gonadotropin releasing hormone. And this gonadotropin releasing hormone stimulates the anterior pituitary gland. The anterior pituitary gland in turn releases two hormones, that is, one is called FSH and one is called LH. Both of them are synthesized in the anterior pituitary gland. Please take down the full form of FSH. FSH is nothing but follicle stimulating hormone. And what is LH? I'm writing it this side, luteinizing Okay, so what are the two hormones released by the anterior pituitary gland? The two hormones which are released by anterior pituitary gland are follicle stimulating hormone and luteinizing hormone. And at the top right corner of the slide, I am writing here for you all, FSH and LH are collectively referred to as, please take down, gonadotropins. All right, so because they have a tropic effect, tropic means to have a stimulatory effect, because they have a stimulatory effect on the gonads, that is the testis in this case, they are called gonadotropins. Now let us see what are the target cells of FSH and LH. The target cell of FSH is Sertoli cell. Okay, and what does the Sertoli cell do? The Sertoli cell helps in sperm production. But I want you to write down what is specifically mentioned in the NCERT to be very precise in terms of our board preparation. Sertoli cells are involved in, I'm writing it here, it is involved in production or release of factors that help in, I hope you all are with me in this, that help in spermiogenesis. All right, so if there is a PU question, don't write that it helps in sperm production, which is a very loose or a less specific way of saying. So be very specific and say that the Sertoli cell, we've already learnt before that the Sertoli cell also provides nutrition to the male germ cells and the newly formed sperm. That is one thing. And what does FSH specifically do to the Sertoli cell is the question here. FSH acts on the Sertoli cell and the Sertoli cell helps in the production. In turn, it releases or produces certain factors that help in spermiogenesis or spermateliosis. Is that done? All right. So now LH. Let's move to LH now. What does LH do? LH acts on which cell? Leydig cells, which are present outside the seminiferous tubule. And the Leydig cells, again I'm going to mention what is written in the NCERT here. The Leydig cells synthesize and secrete hormones called androgens. Androgen synthesis and secretion. All right, and by now you know that it's mentioned here anyway. One of the principal members of the androgen family is testosterone. So don't use the word testosterone as much as possible. In brackets you may write so, but you have to write androgens. Androgen is the male sex hormone. The principal member is testosterone. Is it clear to all of you? The process of hormonal control. Hypothalamus. So which are the endocrine glands involved here? Can you tell me? Okay, hypothalamus, yes. And I hope you know hypothalamus is a part of your brain. Then anterior pituitary gland and testis. See, surprisingly, the testis is also an endocrine gland, because it has two cells which produce hormones. One is Sertoli cell and one is Leydig cells. Okay, so hypothalamus secretes GnRH. GnRH targets anterior pituitary, stimulates the production of gonadotropins FSH and LH. FSH, the target cell is Sertoli cell, which stimulates the production of factors which help in spermiogenesis. And LH, the target is Leydig cells, which help in the production of androgen, the principal androgen being testosterone in the male body. And what do we call FSH and LH collectively? FSH and LH are collectively referred to as gonadotropins. Okay? So this is about the hormonal regulation of the process of spermatogenesis, which they may ask you to write the schematic. However, it's not mentioned it's not given in the NCERT, the schematic representation. If at all they ask you this, it may be a 3-marker question of some of the hormones which have a direct influence on the testis. Alright, so assuming you're all thorough with this slide, now the next one we are going to discuss is the structure of the human sperm. A very important 3-mark diagram question. Alright, so I will be drawing this diagram for all of you, and I want all of you to draw along with me. But before I start drawing, I'm going to show you the diagram which is there on the slide and explain what are the parts of the sperm. So what are the 4 major parts of the sperm? Now all of you look at the diagram, the picture on the right side. So there is head. It has a sperm is nothing but a cell, right? It has a nucleus. It has very little cytoplasm inside it. So the first part of the sperm, the most important part, is the head. The second part of the sperm is a very constricted region which is referred to as the neck. The third part of the sperm is the middle piece, and the last and the final part is the tail. Now why am I numbering these parts? Because in your board exam, they may ask you a 3-mark question. Just mention the parts of a human sperm. So don't have to explain anything. You just have to mention head, neck, middle piece, tail. That's it. You'll get 3 marks. Did you understand? So these are the parts of the sperm. A sperm, like any other cell, is covered on the outside by a plasma membrane. See, the outermost lining is the plasma membrane. Okay. Now sitting inside the head of the sperm is this huge, actually it is a tiny structure. It's a condensed version of the nucleus. This is the nucleus that is located in the head, and it contains chromosomal material. And by now I am assuming all of you know that this nucleus has how many chromosomes? 23 chromosomes. It has only 23 chromosomes because the sperm nucleus is it haploid or diploid? Yes, you're right. It is haploid. Okay. Now, can you all see a cap-like thing sitting on top of the nucleus? Now, this cap-like thing that is sitting on top of the nucleus is referred to as the acrosome. The acrosome contains certain enzymes, because you all know that during fertilization process the sperm has to approach, it reaches the egg, and in order to pass on its genetic material it has to dissolve the egg membrane, it has to break down this egg membrane, and that requires a lot of enzymes that is provided by acrosome. Okay, and all of you please remember that acrosome is derived from the Golgi apparatus of the spermatid. So where does the acrosome come from? The acrosome is a modification of the Golgi apparatus, and it is sitting like a cap on the head, as in on the nucleus of the sperm. Now look at the neck region. Now in the neck region, if you were to be even more precise here, now if this is the head and if this is the neck region, actually you will find, and if this is the nucleus, let me shade the nucleus here, you will find two centrioles. One is a centriole which is very, very closely found to the nucleus, and the other one is a little far behind from the first one. This one, which is closer to the nucleus, is called the proximal centriole, and this one is referred to as the distal centriole. So two centrioles are located in which part of the sperm? The neck part of the sperm. Clear, no? Fine. So now let's move on to the third part, the middle piece. Here they have mentioned the middle piece has two components. One is called the axoneme and the Nebenkern. I want all of you to focus on this rod-like structure, which is the backbone of the sperm. And this rod which I am showing you here, this rod-like structure is nothing but the axoneme. Alright, so it forms the backbone of the sperm. What is Nebenkern? Now can you see these mitochondria here? They are arranged in a spiral. See, these rounded parts are the cut portions of the mitochondria, because we are showing you the internal structure. And can you see that the mitochondria is like a ribbon? It's flat and it is coiled around like this in a zigzag manner, in a spiral manner, and this spiral mitochondria is referred to as Nebenkern. So I'm going to write here, it is the spiral mitochondria. Okay, why should there be mitochondria here in the middle piece? Because mitochondria provide energy source. 1 mark they will ask you, energy source for swimming. So name the structure or the part of the sperm or the component of the sperm that provides energy for the sperm motility. The answer is mitochondria. And what is the name given to mitochondria? Nebenkern. Kern means nucleus, kernel. Neben means near. So it's near the nucleus. That's why it is called Nebenkern. Alright. Fine. The last part of the sperm tail is of the sperm structure is the tail. In the tail, can you notice this portion, the thicker portion, is referred to as the principal piece or the main piece, and this only thread-like portion that you see here is referred to as the end piece. So main or the principal piece and the end piece together make up the tail or the flagellum, and this tail of the sperm, it is the one that helps the sperm to swim rapidly in the female reproductive tract onwards to its female gamete. Is that clear to all of you, the structure of the sperm? Now I want all of you to be ready with your pencil and your book, so that in the next slide I'm going to draw the structure of the human sperm, because this is a diagrammatic question. I'm going to simplify the diagram with all its details intact. Okay, so let's move on to the structure of the human sperm. So first I'm going to show here the head of the sperm. Okay, this is the head of the sperm. Then I'm going to show a small constricted region, which is the neck, and then I'm going to show this portion, the main body or the middle piece of the sperm. Okay, and again I'm going to make a constriction here. Fine. So now let us fill up the head. Now in the head what did you see? You saw a prominent nucleus. So let me shade the nucleus like this. Sitting on top of the head, you saw a cap-like structure. This cap-like structure, let me put some dots inside, is the acrosome. In the neck, I told you there is a centriole behind the nucleus, immediately behind the nucleus, and a centriole away from it. Correct? So the centriole behind the nucleus is the proximal one, away from it is the distal one. Now arising from the distal centriole is the backbone of the sperm. See this rod-like structure I was talking about. And then I'm going to make it a more narrowed portion, and then let me elongate the same like this. This is the axoneme. All right. Okay, now I'm going to fill up the middle piece with mitochondria. So let me show these circles which represent the sections of the mitochondria. And how do I show that it is ribbon-like? So let me connect these sections to give you a... It shows that it is a ribbon-like spirally coiled mitochondria. And let me put some zigzag lines inside, because you know that mitochondria has cristae and all of that. So we are done with mitochondria in the mid piece. Now here, where I've shown a constriction like this, I'm going to show a ring-like structure. I'll come to what this is. And then the initial part of the tail has a covering on it like this. I'm going to stop the covering here. So the remaining part of the tail is end piece. Wherever the tail is covered by this structure, which is shown over here, the plasma membrane, that is called the principal piece. I will come to the difference much later. Now let us label the parts, okay? Yeah, so first what am I supposed to label? I'm supposed to label the plasma membrane. Don't forget that it is a cell. It is a cell which is covered by plasma membrane. Then let me put a flower bracket here and write head. Head of the sperm. Then what is this cap-like structure sitting on top of the nucleus? Let me label that. It is the acrosome. Then what is this shaded structure? The compact oval-shaped structure. This is the nucleus. Containing what? Chromosomes. N is equal to 23. Okay. Then what is this structure? This narrow constricted portion here. This is the neck. In the neck, what are the two centrioles? The one behind the nucleus, immediately behind, is called the proximal centriole. The one farther away from it is called the distal centriole. So I'm going to cover these two and write them as centrioles. Done. Then what is this part of the sperm body referred to as? This is referred to as the middle piece, or simply the mid piece. What is the backbone of the sperm called? Axial filament. Or axoneme. What are these ribbon-like structures which are spirally coiled? They are nothing but the mitochondria. In brackets you write energy source for swimming. Correct? Fine. Now, I told you that there is a ring-like structure here. This is another ring. It's actually another centriole, which is not there in your textbook. So, I'm just giving this for your information. It is called the Jensen's ring, or it is simply called the ring centriole. okay, whose origin is not known entirely. And then you can simply, like in the textbook, you can simply label this portion as the tail. To be very precise, you may label this part of the tail as end piece, and this covered part of the tail as main piece. Now, what is the difference between end piece and main piece? Okay, so let me just, this is the backbone of the tail, the axoneme. So in the difference between the end piece and the main piece is, now if this is the main piece of the tail, remember I'm showing you the tail here to be very, very specific. So this is the main part of the tail and this is the end part. Notice that this is the plasma membrane, okay? So what I'm showing you here, the outer covering is the plasma membrane of the tail. Now it is not like shown here that it's not that the end piece doesn't have a plasma membrane. The end piece, see how the plasma membrane hugs the axoneme in the end piece. It's very, very closely attached. And here in the main piece, the gap, there is a gap between, there's a lot of space between the plasma membrane and the axoneme. But here you can see how the plasma membrane has literally sheathed or hugged the axoneme, and this is why this is referred to as the end piece. So this is one of the differences. Why there is so much gap between the axoneme here, between the plasma membrane and the axoneme, is because there is a special fibrous protein, longitudinal fibers, which extend just inner to the plasma membrane. Okay, so there is a fiber, there are two longitudinal fibers which extend, and they are made up of special proteins and which extend between the plasma membrane. This is the plasma membrane and this is the axoneme. So between the axoneme and the plasma membrane there is something called the fibers, or you can say longitudinal fibrous sheath. So this fibrous sheath is missing in the case of end piece. Okay, this is just for your knowledge, just for your information. I want all of you to mostly focus on this part of the sperm structure, because this is what they expect in your board exam. All right, so we have now replicated, this is just a very, very rough diagram, as much as I could manage using this apparatus that I have with me. I'm sure you could be much, much neater when you write it on your book. Okay, so all the parts have been labeled. However, when the question is asked in your PU exam, you may ignore the Jensen's ring or the ring centriole. You may ignore the centrioles, like the diagram that I had shown in the previous slide. Okay, so this is about the structure of the human sperm. I hope all of you have managed to write it in your books and you have followed the way I have drawn the diagram, so that it is easy for you to replicate the same in your board examination. I want all of you to practice this diagram well, because it is commonly asked for a 3-mark question. All right. Last but not the least is we need to discuss about something called semen. And during the time of sexual intercourse, or during the time of coitus, the human male releases or ejaculates about 200 to 300 million sperm into the female reproductive tract. All right, so this is quite a huge number. 200 to 300 million sperm are liberated, are released, are ejaculated into the female reproductive tract, and they start their journey onwards, upwards the female reproductive tract into the fallopian tube. For a person to have normal fertility, he must have at least 60% of his sperm with normal shape and size, and at least 40% of the sperm should show vigorous motility. Clear? And the sperm are released from the seminiferous tubule. Where do they go once the sperm are released from the seminiferous tubule? They go to the accessory ducts. You've learnt about it in the previous lecture. They pass into the rete testis, then vasa efferentia, then epididymis, then vas deferens, and then they ascend up into the abdomen, behind the urinary bladder, ejaculatory duct, urethra, and finally to the outside, isn't it? Then we should learn to distinguish between these two terms. One is seminal plasma and one is semen. What is the difference between the two? The secretions of all the male accessory ducts, not only the glands, secretions of epididymis, secretions of vas deferens, secretion of seminal vesicle and the prostate gland, collectively these secretions are referred to as the seminal plasma. But once the sperm are added to the seminal plasma, along with the sperm, the seminal plasma along with the sperm is referred to as the semen. So what is ejaculated into the female reproductive tract is not just the seminal plasma, it is the sperm suspended in the seminal plasma. Okay, so that suspension of the sperm in the seminal plasma is what is referred to as the semen. So this is a question you can expect. Distinguish between seminal plasma and semen. Alright, so please go through this slide very thoroughly and understand the difference. Seminal plasma is not just the secretion of the gland, it is also including the secretion of epididymis and vas deferens. And when the sperm is added into this, or the sperm gets suspended in this from the testis, the new sperm are produced and they are sent out of the seminiferous tubule. And once there is sperm within the seminal plasma, we call it as the semen. Clear? No doubts? Alright. So what did we learn today to wrap up this particular lecture? We learned about the general meaning of gametogenesis, formation of sperm in the testis, and formation of ova in the ovary in male and female respectively. Yes or no? Correct. Then we learnt about spermatogenesis. One of you close your eyes and recall the definition of spermatogenesis. What is it? It is the production of sperm from immature male germ cells or spermatogonia in the seminiferous tubules of the testis. And when does it start? At puberty. Good. What is spermiogenesis? Transformation. Who transforms into who? Yes, you're right. So there is a spermatid which transforms into a motile sperm, and this transformation is called spermiogenesis. Good. What is the alternate term? Yes, spermateliosis. Good. So spermiogenesis is also called spermateliosis. And we also added a short explanation on something we had learnt in the previous lecture. Spermiation, that is the release of the sperm from the seminiferous tubule into the rete testis. Then we learnt about hormonal regulation. What were the hormones? GnRH by hypothalamus, FSH and LH by the anterior pituitary gland. Who does FSH act on? Who does LH act on? I will let you find the answers. You can go through the videos once again and you can learn the answers and you can read from the textbook and be prepared for your upcoming homework, okay? Then we learnt about the structure of the sperm. Diagram question I told you very clearly. And then we learnt about semen and how to distinguish between seminal plasma and semen. So these are some of the things which we have managed to learn in today's lecture. And then what's the next thing? What's the usual drill? Yes, you're right. Homework. Without homework, whatever you learn is highly volatile and it is very easy to forget the same, isn't it? So I want all of you to take your homework extremely seriously. Sit with your textbooks, sit with the videos that we've made for your benefit and go through them again and again and again. It's already uploaded. You can go through them n number of times. Be thorough with the concept and please do your homework. Trust me, indulging in some activity like this will also keep your mind distracted and your mind healthy, isn't it? Okay. Then, the first question is, name the target cells of FSH and LH in the testis. The second question is, The second and the third questions are definition questions. Define spermatogenesis and define spermiogenesis. I want all of you to take down the questions of the homework. Okay. First question is, name the target cells of FSH and LH in the testis. You can pause the video in between and take it down. That's not an issue. Define spermatogenesis. Define spermiogenesis. Fourth one is mention the different parts of the human sperm. See, I told you, you just have to mention the parts. There are 4 parts. I'm giving you a hint over here. There are 4 parts which you need to mention. Last one, which I just discussed in the last but one slide. Distinguish between seminal plasma and semen. Seminal plasma plus dash is semen. See, I gave you a hint. Your homework is all set. There are only 5 questions and you need to be thorough with these 5 questions. It is so simple. You will get additional homework later. Clear? And even though it is not asked in the homework, I want all of you to be thorough with schematic representation of spermatogenesis. All right. So with this, I will be wrapping up this session with you all. I hope you all understood the concept of gametogenesis and spermatogenesis. And all of you have a wonderful time. Happy studying and happy homework doing. And also take good care of your health and stay at home. And be cheerful, be happy, think happy thoughts. All right. So we'll see you until next time. Take care and bye.